Welcome to the deep dive where we really try to cut through the noise and get you the insights you need.
Great to be here.
So today, we're plunging into a really significant challenge, one that impacts millions across the globe: lung cancer.
It's a devastating one.
It really is, often because it's such a silent threat,
Yeah.
In those early stages, it's incredibly hard to catch. And globally, it's still a leading cause of cancer deaths.
Yeah, the numbers are stark.
And we all know or hear about the promise of early detection. Catch it sooner, the better the outlook, generally. But the standard methods, things like low-dose CT scans, well, they have limitations.
Right, false positives are a real issue.
Exactly. False positives, the anxiety that comes with them, and then, this whole cascade of follow-up tests that might not even be necessary.
It's a difficult balance.
So that leads us to, I think, a really compelling question. It almost feels a bit sci-fi. But what if? What if we could detect cancer, specifically lung cancer, non-invasively, like with just a simple blood draw? And not just detect it, but do it with, well, much better accuracy than we currently can.
Well, it's moving out of sci-fi and into reality, and that's the exciting part. We're talking about EDGC's OncoCatch S service.
OncoCatch S, okay.
Yeah, it's positioned as a really revolutionary answer to that exact challenge you just laid out.
Right.
The core idea is right there in the name, essentially. Early detection of cancer with a single blood draw.
A single blood draw. That's the hook.
That's the goal. It's really designed to fundamentally shift how we think about cancer screening and eventually diagnosis.
Okay, let's dive in then. What we really want to unpack today is the science, the genius maybe behind OncoCatch S.
Let's do it.
How is it using this cutting-edge tech to achieve this detection? And maybe most importantly, what could this actually mean for you listening out there? For the future of screening, for personalized medicine.
Big questions.
Yeah, our mission is to leave you with a really clear picture of how this kind of innovation could genuinely transform healthcare.
Sounds good.
So let's start at the beginning. The foundational concept, liquid biopsy. Now, you hear this term a lot more lately.
It's gaining traction.
We're mostly familiar with traditional biopsies, right? Where doctors physically take a piece of tissue. Can be pretty invasive.
Definitely. Sometimes difficult, depending on the location.
But a liquid biopsy, that's different. Imagine your bloodstream carrying not just, oxygen and stuff, but also microscopic clues about your health.
Tiny messages.
Exactly. A liquid biopsy analyzes cell-free DNA, cfDNA. These are like genetic whispers, tiny bits of DNA released into the blood. And the crucial part is some come directly from cancer cells.
That's the key insight.
And the beauty of it, the advantage for OncoCatch S, no need for that invasive tissue sample.
Precisely. That's a massive benefit. OncoCatch S takes the cfDNA, these genetic whispers, and processes it using next-generation sequencing, NGS.
NGS, okay. High-tech stuff.
Very high-tech, combined with really advanced bioinformatics. It's not just simple lab work, it's complex analysis. It lets them really meticulously look at the cfDNA from cancer cells to find specific genetic changes.
So finding the mutations, the errors.
Finding mutations, alterations. But what's really interesting is how this detailed analysis aims to go beyond just yes cancer or no cancer. It's about providing personalized guidance based on those unique genetic findings. It's like getting a molecular fingerprint of that specific cancer.
Molecular fingerprint, I like that. So it's not just a binary outcome. And thinking about the practical side, beyond avoiding the tissue sample, which is huge, what about these other benefits mentioned, like minimizing interpretation and minimizing deviation? What do those mean for me if I were taking the test?
That's a good point. They sound a bit clinical. Minimizing interpretation and deviation. It basically means the test is built for consistency and reliability.
Yeah.
It standardizes the analysis so the results are as objective and reproducible as possible. Doesn't matter as much who runs the test or reads the raw data. For the patient, for you, that means highly consistent, dependable results you can trust.
Trust is key, obviously.
Absolutely. And yes, it aims to go further by providing a detailed description for cancer treatment. That's incredibly valuable. It's not just detection, it's providing insights to directly inform the next steps, guiding personalized treatment right from the get-go.
That really changes the flow, doesn't it? From finding it to figuring out what to do about it. So based on what we know, how is OncoCatch S structured? Are there different versions or options?
Yes, exactly. The sources suggest specific test options. First, there's cancer screening.
For healthy people.
Well, for asymptomatic people. It's designed specifically to detect and analyze cancer-related mutant genes in asymptomatic patients. This hits that crucial need for early detection before symptoms show up.
That's the dream scenario, really.
It really is. Imagine catching cancer at stage I, when treatment is often much more effective. Then, for patients who already have a diagnosis, there's companion diagnostics, or CDX, for therapy.
CDX, right.
This provides the basis for personalized medicine prescription. So it helps doctors choose treatments tailored to that individual's specific cancer profile based on its genetics, moving away from that one-size-fits-all approach.
Towards targeted therapies.
Precisely. And just briefly, cancer monitoring is also mentioned as a potential use, maybe for tracking treatment response or recurrence later on, but we'd need more details on that specific application.
Okay, that makes sense. Now, you mentioned combining data, getting that fingerprint. This feels like where it gets really interesting, this idea of multi-omics.
Ah, yes, multi-omics. It's a powerful concept.
Can you break that down? Why isn't looking at just one thing, like one gene, enough for something as complicated as cancer?
That's the perfect question. Multi-omics is essentially about integrating multiple layers of biological information. Think of it like investigating a complex scene. You wouldn't just rely on footprints.
Yeah, you'd want fingerprints, DNA, maybe security footage.
Exactly. You combine all the evidence, fingerprints, DNA, witness reports. Each piece tells you something, but together you get a much clearer, fuller picture. Cancer is incredibly complex. Looking at just one data type, maybe one gene mutation, often doesn't capture the whole story.
Okay.
Multi-omics lets us see how different biological systems, different homes are interacting. It gives a more holistic and ultimately more accurate view of what's going wrong.
So what specific omes or data types did the EDGC study actually pull together for their lung cancer test? What were the pieces of that puzzle?
Right, so in their study, the one published in Cancers in 2023, they integrated 3 main types of data. First, blood tumor markers.
Like proteins in the blood.
Exactly. Specific proteins that can indicate cancer activity. They measured 5. Cypher 21-1, CA15-3, AFP, CEA, and CA19-99. And what they found was telling significantly higher levels of 4 of those, Cypher 21-1, CA15-3, CEA, and CA19-9, in the lung cancer patients compared to the healthy controls.
And some of those are already used, right?
Yes. Cypher 21-1 and CEA are actually commonly used for lung cancer diagnosis, so seeing them elevated wasn't a total surprise, but it was a key piece.
Okay. What was the second data type?
The second was cell-free DNA, cfDNA concentration. Basically, just the total amount of this free-floating DNA they could extract from the blood sample, a quantitative measure.
So how much DNA is floating around? Got it. And the third?
And third, they looked at copy number variations, or CNVs. Now think of our DNA like a huge instruction manual. CNVs are when sections of that manual get accidentally copied multiple times or sometimes deleted altogether.
Extra copies or missing bits.
Exactly. These changes in the number of gene copies are really common in cancer cells. And crucially, they can detect these CNVs from the cfDNA using that NGS technology we mentioned earlier.
Wow. So you've got proteins, the amount of DNA, and these structural changes in the DNA itself. 3 different angles.
3 different angles, all providing different kinds of information.
So pulling it together, if you have these 3 distinct data types, how does that translate into a more reliable diagnosis? Because you said earlier, one type on its own might be a bit weak. Is that where the machine learning comes in?
That's exactly it. You nailed the crucial point. While each piece, a single blood marker or just the cfDNA amount, is useful on its own, it often has, as the study puts it, relatively low discriminative power.
Meaning it's hard to tell cancer from non-cancer reliably.
Right. It's like trying to identify someone just by, say, their hair color. Helpful, maybe, but not definitive. This is precisely where machine learning ML becomes absolutely essential.
The AI helper.
Sort of, yeah. Machine learning is a powerful tool designed specifically for analyzing these huge, complex, multi-layered data sets. It can find patterns that humans or even traditional stats might miss.
Right.
ML algorithms sift through all this data, the tumor markers, the cfDNA levels, the CNVs looking for subtle, complex patterns and anomalies that signal disease, even patterns that aren't immediately obvious. The EDGC study used specific ML algorithms like adaptive boosting, AdaBoost, multi-layer perceptron, MLP, and logistic regression, LR. They essentially learned from the data to build predictive models.
To separate the healthy from the cancer samples.
Exactly, to make that distinction much more accurately.
And the results they published, They really seem to back this up, showing the power of combining everything.
They really do.
They found that even when they just combined two key blood markers, Cypher21 and CEA, the accuracy jumped significantly. Now, for listeners, accuracy is often measured by AUC, area under the curve. 1.0 is perfect. 1.5 is random chance.
Right.
With just those two markers, the AUC got up to 0.828. That's pretty good already.
It is definitely a good starting point.
But, and this seems to be the clincher, the real breakthrough came when integrating multi-omics data into ML significantly improved accuracy. When they combined all three data types, the markers, cfDNA concentration, and the CNVs.
That's when it got really impressive.
The AUC value shot up, reaching as high as 0.931 with one of the models, the MLP model. That leap from around 0.83 to over 0.93. That's substantial.
It's a huge improvement. It demonstrates what the study calls exceptional discriminatory ability. It really shows that when you feed the machine learning all those different clues together, it can build a much, much clearer picture.
That clarity is key. And that jump in accuracy really is striking. Now let's talk about something super critical for patients. Can this multi-omics approach detect cancer at different stages? Because early detection is the name of the game, right?
Absolutely. That's arguably the most important aspect. And yes, the study looked into this specifically. While using just single markers or cfDNA concentration alone showed only, let's say, modest accuracy in the very early stages.
Which is the hardest part.
The full multi-omics integration analyzed by ML provided remarkably high AUC values across all stages. For stage 1, those earliest cancers, the AUCs were really promising between 0.797 and 0.874.
Almost 0.8 to nearly 0.9 for stage 1. That's strong.
Very strong for stage 1. And then as the cancer progressed, stages 2 and 3, the AUCs climbed even higher, consistently over 0.9. And for stage 4 advanced cancer, they were exceptionally high, actually exceeding 0.966.
Wow, almost perfect discrimination for late stage.
Extremely high. Now, to put some sensitivity numbers on that, sensitivity being the ability to correctly identify those with the disease at a set specificity, like 90% to limit false positives.
Okay.
For the multi-omics approach, stage I sensitivity was in the range of 50% to 68.2%. Stage 2 jumped to 80-90%. Stage 3 was around 65.2, 73.9%. And stage 4 had the highest sensitivity, hitting 89.7% up to 96.6%.
So pretty good detection rates, especially stage 2 onwards, and decent for stage I too.
Yes, it shows significant power across the board, offering a real chance for earlier detection while also being very effective later on.
So, connecting this back to you, the listener, why should you care about these AUCs and sensitivity percentages?
Right, this so what?
Yeah. This high accuracy, especially its potential in early stages, but also its strength later on, it means earlier, more precise diagnoses are genuinely becoming possible. And that directly translates to potentially much better treatment outcomes, better therapies chosen, and ultimately improved survival rates.
It changes the whole trajectory.
Imagine detecting lung cancer when treatment is often less harsh and more likely to succeed. It's about shifting from reacting late to proactively finding it early.
Absolutely. Now, it is important, as with any new science, to be transparent about the limitations mentioned in the study itself.
Of course. Good science acknowledges limitations.
Exactly. The sample size, for instance, was relatively small, 92 lung cancer patients, 80 healthy controls. Promising results, yes, but this means more validation, external validation with larger, more diverse groups of patients is absolutely essential to really confirm these findings.
You need to make sure it works broadly.
Precisely. And we also have to acknowledge the complexity. Integrating all this diverse data, running these sophisticated ML models, it's computationally intensive. It requires significant resources and expertise. It's not a simple dipstick test, you know.
Right, it's complex, cutting-edge science. Those are fair points.
Yeah.
And while this study focused specifically on lung cancer, the potential feels broader, doesn't it?
Oh, definitely. The underlying principles, multi-omics data integration, using machine learning to find patterns, these could theoretically be applied to other cancer types too.
Makes sense.
Of course, every cancer is different. Each would need its own specific biomarker research, its own tailored models. But the foundational approach, the methodology, is clearly showing incredible power here with lung cancer.
So let's try and sum up our deep dive today. It feels like EDGC's OncoCatch S represents a really significant step, maybe even a leap, forward in cancer detection.
I think that's fair to say.
It's non-invasive, just a blood draw. And it harnesses this combined power of multi-omics data, different biological clues, and smart machine learning to get highly accurate results, particularly shown here for lung cancer. Feels like more than just a small improvement.
Agreed. It feels like a potential game changer. What's truly exciting is the promise it holds for the future. It moves us closer to more effective, more personalized,
And less invasive.
and less invasive cancer therapies. Predicting treatment response better, boosting treatment efficiency through earlier, more precise detection. OncoCatch S really does offer a tangible path towards transforming oncology, making treatment more targeted, more successful.
So as this technology develops, as it gets more validation, here's a thought to leave you with. How might a regular, non-invasive deep dive into your own biology through tests like this change how we approach health monitoring? How might it change proactive disease prevention for you, for your family, in the years ahead?
It really could reshape preventative medicine.
Something powerful to think about. Thanks for joining us on the Deep Dive.